Neuropeptide Y promotes adipogenesis in chicken adipose cells in vitro.
So for a little background information, neuropeptide Y is a 36 amino acid neuropeptide that acts as a neurotransmitter in the brain. It is also known to enhance food intake in vertebrate species and promotes adipogenesis in mammals. Adipogenesis is the process of cell differentiation by which pre-adipocytes become adipocytes, which are also known as fat cells, which is a cell that stores fat and connective tissue. The objective of this study was to determine if neuropeptide Y also enhances adipogenesis in birds, using chickens as a model. We know that with the injection of NPY, it stimulates food intake. In this study, we tried to determine the effects of NPY on chicken adipose tissue function. We know that there is a major concern in the poultry industry about the control of body fat accumulation, and by using chickens as a model, hopefully we can understand the pathogenesis of obesity in humans. For experiment 1, neuropeptide Y dose/treatment was of 1 nanomole compared to 0 as a control. Cellular proliferation was evaluated to study the effects of NPY, and to evaluate adipocyte differentiation, we looked at oil red O staining, glycerol 3-phosphate dehydrogenase specific activity, and mRNA abundance. Each differentiation experiment was evaluated at 2, 4, and 6 days post-induction. So this graph shows how NPY affects gene expression of proliferation markers. For the treatment of the cells with the 1 nanomole of NPY, it did not have an effect on cellular proliferation, although we did see one difference with this dose with an increase in neuropeptide Y receptor subtype 2 mRNA abundance. The first figure shows oil red O staining. The red color indicates the staining of neutral lipids, and we can see that by day 4 there is a distinct lipid accumulation in the cells. The second figure shows the treatment of the cells with NPY shows an increased glycerol 3-phosphate dehydrogenase activity on day 6 post-differentiation. These two figures show the mRNA abundance of proliferation and pre-adipocyte markers in neuropeptide Y treated cells. The first figure shows that treatment with the NPY increased the abundance of thyrodoxine-dependent peroxidase 2 mRNA at day 4 and day 6. The second figure shows that treatment with NPY increased the abundance of GATA-binding protein 2 mRNA at day 2 and day 6.
Within experiment 2, there was a neuropeptide Y treatment of 10 nanoparticles. Then a fetal bovine serum was used for the proliferation of the cells. After the proliferation of the cells, adipocyte differentiation was applied. There was then an oil red staining, and lastly G3-PDH specific activity. The results showed that treatment with 10 nanoparticles NPY was associated with an increase in NPY2R mRNA abundance at 4 hours post-treatment in comparison to non-treated cells during proliferation. Treatment of NPY during adipocyte differentiation reduced regulatory element binding protein on day 4. Increased fatty acid binding protein 4 mRNA on day 6 post-differentiation, as well as expression of lipoprotein lipase was greater on day 6 after differentiation, and also an abundance of Ki67 mRNA was greater at day 4 and 6 after differentiation. There was a greater quantities of TOP28 mRNA at day 2, 4, and 6. There was also an increased abundance of TPX2 mRNA at day 4, and a greater abundance of GATA2 mRNA at day 6.
Experiment 3, or Trial 3, looked at a neuropeptide Y treatment of 100 nanomole. Cellular proliferation was evaluated to study the effects of neuropeptide Y on the cells. Adipocyte differentiation was also observed using oil red staining. Glycerol 3-phosphate dehydrogenase activity was also looked at as a part of this trial. 100 nanomoles of neuropeptide Y in vitro injections showed both positive and negative effects on certain gene proliferation markers. For example, there was a significant increase in both neuropeptide Y and neuropeptide Y receptor type 2 fold differences in mRNA. Conversely, the top 2 and TPX2 showed a significant decrease in proliferation, as seen in the top graph to the right. As mentioned before, glycerol 3-phosphate dehydrogenase activity was studied. As seen in the lower graph to the right, there is a significant increase in G3-PDH activity, most notably in day 6, where there is a significant increase in this activity. Additionally, there is a clear sign of growth of lipids when oil red was stained. Treatment of 100 nanomole of neuropeptide Y increased the number of mRNA on both fatty acid binding protein 4 and lipoprotein lipase significantly. Conversely, treatment with 100 nanomoles of neuropeptide Y decreased significantly the abundance of mRNA of both sterile regulatory element binding protein and cat enhancer binding protein alpha. These factors are associated with adipogenesis and indicate its progression. The chart to the bottom right shows the mRNA levels of neuropeptide Y. It is clear to see that neuropeptide Y supplementation has increased the expression of neuropeptide Y on both days 2 and 6 post-differentiation. This possibly suggests that a positive feedback effect is on its own expression in the adipose cells. The chart to the right shows the mRNA abundance of proliferation and pre-adipocyte markers in neuropeptide Y treated cells. There are significant increases in the mRNA abundance of 3 proliferation factors, Ki67, TOP2A, and TPX2. Additionally, there's an increase in mRNA of 1 pre-adipocyte marker, GATA2. As seen in the proliferation factor Ki67, there is a significant increase in the mRNA abundance at days 4 and day 6. In TOP2A, there is a significant increase across all 3 days. In the final proliferation factor, TPX2, some notable increase in day 4 and a great significant increase in day 6. Finally, in the pre-adipocyte marker, GATA2, there is a significant increase at day 2 and day 6.
The treatment with increased neuropeptide Y was also associated with enhanced lipid accumulation, or adipose accumulation, and the expression of adipogenesis-associated factors. Furthermore, tag synthesis was increased due to an increased amount of G3P and G3PDH, which is due to a lack of activity of glycerol kinase within adipose tissue. Tag synthesis shows an indirect marker of increased adipogenesis and is correlated highly with differentiation of the precursor cells to terminally differentiate adipocytes. Expression of the neuropeptide Y2 receptors were not directly affected by the neuropeptide Y treatment, but they did show a decrease in expression from days 2 to day 6, which indicates that they are heavily involved in adipocyte dynamics, and specifically they're regulated during the stages of the adipose life cycle. This was most noticeable at day 6, which is noticed within the following diagram, seeing as the 2 letters are different, which shows a significant difference. Overall, it appears that neuropeptide Y tends to promote adipogenesis in avians through neuropeptide Y2 receptors, mediated induction of the pre-adipocyte proliferation, fatty acid metabolism, transport, and the tag synthesis. These are all marked, particularly around day 6, by a rapid increase in DNA synthesis. So, according to the study, this is the first of its kind to measure adipose in chicks in order to control the accumulation of body fat. So this would be crucial within the poultry business and the control farmers have over their livestock. Furthermore, this study has the potential to become an extension of the understanding of obesity in adult humans. One critique for this study is that more time could have been allotted for the neuropeptide Y to exert maximum efficiency. The study itself indicates that 4 and 12 hours may not have been sufficient time enough, and that the neuropeptide Y exerts maximum efficiency within the first 24 hours, so more time. Moreover, the amount of broiler chicks was not disclosed, meaning that there could have been insufficient samples. Another critique is that there's only one type of chicken used. Could show insufficient results because it could affect different types of chickens differently. Furthermore, it would be more sufficient to transfer this target to humans where mammals used, i.e. rodents. Several positive notes of this study were that each experiment had 3 biological replicates to ensure consistency within the findings. Furthermore, the experiment supplemented the serum-derived neuropeptide Y with chicken neuropeptide Y to limit variability within the testing.